To talk about our experiences with the RDCRN. We were with the initial first cycle. So we've been at this for close to 20 years now. And it's really been just a hugely impactful experience in my life. So I'm happy to be able to share that. First of all, let's talk about what PCD is. So this is a micrograph of cilia. As you can see, they look kind of like hair-like things, almost like sea anemones or something. And these line all the epithelial tissue in your body, which is found in your upper and lower respiratory tract, ventricles of the brain, and the reproductive organs. So when we talk about PCD, we are talking about genetic disorders or variants that change the function of these cilia, the modal cilia. There are 4 types of cilia, and the 2 we're interested in for PCD are modal. They move, and they have a role in moving the fluid around them or moving through the fluid around them. And here's what a cilium looks like. It's not just a hair. There's actually, in this structure right here, 500 proteins that are going to making up a cilium. PCD, we're interested in this part called the axoneme, the part that looks like a hair protruding out of the membrane. And this is made up of roughly 200 proteins. And from what we know, a defect in any of those proteins may result in the clinical disorder we call PCD. The basal body down here, that's what anchors the axoneme, the ciliary axoneme, to the epithelial tissue. There are 300 proteins in this structure, and these are the non-modal ciliopathies. This is where they derive from, and some of those are just very devastating disorders. So we now know that there are more than 50 genes that have been identified to cause PCD. Variants in these genes cause PCD. And they are across the gambit. At first, we thought that it was just an autosomal recessive disease. Now we know that there are actually dominant versions and versions of this that are associated with X-linked or other syndromic disorders. And the important part about this is that these are actually, you could look at this as having as 54 separate diseases, because each of them has their own role, their own phenotype, their own genotype. But what we have done is kind of lump them together into this one disorder category called primary ciliary dyskinesia. And these are the body areas affected. Primarily the respiratory system, both upper and lower respiratory system, but there are also issues with fertility. There are issues, in fact, we just had a paper that talked about normal pressure hydrocephalus that shows up in almost all adults with PCD. And one of the big hallmark features is asymmetry in the body, called situs inversus, situs ambiguous, or heterotaxy. So this is what they look like. They move in unison. The fluid around them moves them. They sweep in unison using a molecular motor called dynein. And that is how we all keep our airways clean. That is the primary host defense for our lungs. The cilia move, they sweep mucus that's got bacteria or debris in it out and up to the upper airway to be coughed out. So here's what it looks like when it works. These are my cilia, by the way. But you can see there's a coordinated beating pattern, and that pattern helps to move cilia or debris up and out. These are my daughter's cilia. She has PCD. And you can see there's an occasional flicker of a cilia or 2 down here, but there's just a complete loss of ciliary function. So the lungs are not protected. But there's another kind of cilia that's also very important in PCD, and this is called a nodal cilium. And you can see these move differently. They don't beat, they move in a circle. And these cilia only appear at one time in our life, and that's in the embryonic period at the point of gastrulation, where the gut and the respiratory tract the heart are dividing. And it is the circular motion of these cilia that tells the heart tube which way to turn, which way to start to develop. And in the absence of that signal, laterality and the development of the heart can be affected. So what do these defects result in? Obviously, laterality defects, that's about 50% of all patients, because without that signal, It is completely random how things develop. Mostly this is situs inversus, where everything is completely reversed. It all works fine, but it's a mirror image of what it should be. But heterotaxy and congenital heart defects are also associated with PCD. There are issues with brain development. It's rare, but we do have, now it appears it's not quite so rare in adults, that normal pressure hydrocephalus is a bigger problem than we realized. These problems are evident from birth. 80 to 90% of children with PCD have neonatal respiratory distress, and usually they work through it, and if they're supported, they can survive it, but it can be fatal in some cases. The chronic severe airway inflammation is chronic from birth. Hearing loss is a part of that from chronic ear infections. But also over time, the repeated infections can result in neurogenic hearing loss, which requires cochlear implants or hearing aids. The pulmonary infections are actually the biggest problem in PCD because they are the thing that causes the life-shortening aspects of this disorder. The airway clearance is completely impaired, so nasty progressive infections happen and they damage the lungs. In fact, this damage, which is called bronchiectasis, is often seen in childhood in PCD. And this can lead to respiratory failure and with early death or the need for lung transplant. Males are infertile and females, not all males, many males are infertile and females may be subfertile, but the use of assistive reproductive technologies can be very helpful. So this was the environment I was coming into in 1983 when my daughter was born. At that time, the syndrome called Carter-Gainer syndrome was well known. It had been published in the 1920s, but the cause for this syndrome, which was situs inversus, lung disease, and sinus disease, was not known until the 1970s. So when she was born, this was relatively new disorder, and she had pretty much every hallmark symptom of PCD. And when she was finally diagnosed 7 years later in 1991, my first question was, okay, where do we go for research? And I was told there never would be any because it was so genetically complex and so rare, there just wouldn't be any value in doing research on it. So that got me thinking, especially because the disorder was so similar to cystic fibrosis, of what the CF Foundation had done. And really what they did was create a network of expert sites and a clinical registry. And that is what moved their disease forward and brought them to the point now where they have just incredible therapies. And so we talked to the CF group and they told us to package this early program of expert sites and a registry into something called the Path to Clinical Trials. So around 2000, this is what we started looking at. And at the time, it was just me and another patient, and we had no idea where to start. But serendipity came into this, and this is about the time, 2000 or 2002, in that time period, that the government actually created the Office of Rare Diseases Research, and an RFA came out for creating research networks. And we were fortunate enough to get, with our clinical and research partners down at UNC, one of these grants. So we have been part of the Genetic Disorders of Mucociliary Clearance Consortium since 2000, I think 2004 is when we had our first patient that enrolled. And this was under the clinical leadership of Mike Knowles and Margaret Lee, both of whom have retired in the interim. And now Stephanie Davis and Tom Ferkel, all of them still from UNC Chapel Hill, are leading the charge. So the GDMCC started with 4 sites, with UNC here in yellow being the main site, and then expanded to 8 sites. And at the time that we started this, we really weren't sure how many patients there were. In fact, we're still not entirely sure. The demographics are a bit all over, but it became very clear that if you built it, they would come. The network had no trouble bringing patients in. So I don't have this slide, but I believe that we saw through the GDMCC networks 1,100 patients from all over the world. So that, I think, surprised everybody because we'd been told this disorder was so rare. So the GDMCC was able to clarify and expand the genotype and the phenotype, enhancing early diagnosis, which diagnosis continues to be a problem in this disease, and helping us to develop standards of care for both diagnosis and treatment. At the time that the GDMCC started, there were 2 known genes. There are now over 50, and this was a dedicated effort with the GDMCC and collaborators overseas to find these genes. And from that, get the first, we were able to create the first clinical genetic test for PCD. Now, genetic testing for PCD is widely available. And we were able to establish the link between PCD and some of the other ciliopathies that we were aware of, but we didn't really know how the cilia all work together. And also the role of congenital heart defects in this disorder, that children born with PCD are 200 times more likely to have congenital heart defects than in the general population. But what this really did was provide a model and a springboard for the creation of the PCD Foundation Clinical and Research Network and our PCD Foundation Registry. So we learned from the GDMCC that there was interest, and with that we have expanded to now over 40 sites that are PCD clinical centers. You can see we divide them here by PEDS only sites. It's still very PEDS predominant, but white is the PEDS and adult sites. And here are the sites we'll be accrediting in the very near future. And these are the sites that are currently participating in our registry. One of the accreditation roles with PCD Foundation sites is that you have to agree to participate in the registry. So hopefully all sites will be participating in time. So what were the benefits of being part of the RDCRN, and what did we learn? I think the main thing that I feel like I learned is just show up. I mean, I had no idea what I was doing most of the time. I didn't really understand a lot of what was being discussed, but I was there. I made sure I was there at the meetings. I made sure that if opportunities came up, that I could jump in and take advantage of them. And it was really a great way to learn and to be a good partner. So if you want to partner well with clinical people and research people, you need to know your stuff. You need to understand your disease, and you also need to understand the environment in which you're operating, because some things that make sense to us as patient advocacy groups won't work in a highly regulated environment that we work in. So it's kind of on us to know what we're doing, and in that way become a reliable and credible partner for our research partners. And then to take advantage of the incredible depth of knowledge in this network. One of the things that struck me very early on is that a lot of my colleagues in this network had very different disease pathophysiologies going on. There was no connection in that regard for our diseases, but we all had similar operational and organizational issues that we were dealing with. And it was really wonderful to learn from each other, and helped us grow and hopefully help them grow. And I just want to mention a few people who are no longer part of the network, but who were extremely helpful to me early on. Chuck Mohan, who was with United Mitochondrial Diseases Foundation, Cindy Lermans, who has tragically passed away with Urea Cycle Disorders Consortium, and Joyce Coleman, who was with the Vasculitis Research Consortium. These people were a sounding board, along with several people that are still involved in this network, but you know, a shoulder to cry on even when things got tough. So take advantage of those relationships if you can. So what is next for us? Talking about sustainability. This has been challenging. I think we're all kind of concerned about this. What we have decided to do is to create what we're calling the PCD Foundation GDMCC Clinical Research Network. So we want to merge the clinical network that we have created as the PCD Foundation with the research network that the GDMCC has created. And probably the goal would be that we have UNC, who's kind of the lead site, operate as the DMCC essentially of this new network that will be focused on clinical trials. Well, clinical research trials and other trial or other research. So the goal here, and this has been borrowed heavily from our colleagues overseas, that we want to be able to offer a one-stop shop for anybody who's interested in doing research in PCD. We want to de-risk it. We want to make sure that they get the best advice that they can get. that we have patient advisory councils as well as clinical people who look at the designs of the of proposals that come in and can make recommendations. We'll work closely with our registry and be able to hopefully, because we still have very few patients, match patients to trial opportunities. So I think this sounds great. We're very excited about it. We haven't figured out how to finance it yet, but that's kind of the next hurdle. So I think that's it for me.
Thank you, Michele. That was a really great overview from diagnosis, starting up from the ground zero, and developing the collaborations, and incredible that the opportunity of the first round of the RDCRN the timing was just a fit for you guys to join together and reach out for. It seems that really helped push you guys into a whole new realm of being able to expand, create reliable diagnostics, and look toward future therapies. So awesome. Thank you for sharing. Does anybody have any questions for Michele? Kristen?
Yeah. Hi, Michele. It's Kristen Whedon. First of all, excellent presentation. Thank you so much. As Raj just said in the chat, wowed by you and all the work you've done over the years. Definitely look up to you, and we'll be picking your brain more. I'm wondering on the I'm sorry, I'm not feeling really well, so my voice is a little rough. With the sustainability, I understand that at the end, you said you haven't figured out how to finance it yet. A deeper question, do you know how much and don't tell me a number, but, or you don't need to, but do you know how much you need to finance it? Have you gotten that far?
We have a little bit. I mean, like any budget, it's a projection, but, the number's fairly astounding and probably not within our grasp right now. So we're looking actually creating a campaign to try to raise the funds to sustain this effort for the first couple of years. But that'll be challenging. I think we've worked together so well for so long that we'll muddle through until we have the funding. But yeah, that's a concern.
You'll find a way. Thank you.
I would say we're talking millions, not thousands, just if that's helpful.
Annually. Yeah. Thank you.
Any other questions?
Hi, this is Raj. I have a quick question. So, Michele, when you guys first came together, was it, your organization working with one particular group of researchers at, I believe you said UNC, and then when this grant came about, when you guys heard about this, did they then reach out to other sites, or how did that even come together? The group that then became the consortium.
Yeah, it was one site. There was one site in the country doing research on PCD, and I believe they had actually started doing that to have a disease control for the cystic fibrosis work they were doing. So I contacted them to get my daughter involved in research there in 1999 or so. And then this opportunity came up, and I had been in pretty much constant contact with that group. And so we jointly pursued this opportunity, and they had, because of their CF work, a nice network already established that they could pull from. So they brought in four sites to help with the PCD and CF studies that we do through the GDMCC. What I would say is we were in constant contact for the past 20 years. We've been talking to each other, at least every 2 weeks, and often, far more often than that. So I think that is a big part of having a really fine-tuned consortium, is that we work kind of seamlessly. And I think that that's been a bit of a surprise to some of the pharmaceutical people that have come into our orbit, is they don't understand that if they talk to us, they're talking to the researchers, and vice versa, because we don't keep secrets from each other on that stuff. So it was, not knowing a lot of people to begin with, but then working very closely for a very long time that helped grow it.
Thank you, Michele. We're going to talk a little bit more about that toward the end of the agenda, the question that Raj brought up. But Paul, do you have a question for Michele?
Yeah. Hi, Michele. Thanks. I think that was a great talk. You mentioned in there that you're interested in trying to match patients to trials, and that's something that we at the ALS Association are working on as well. And I was just wondering if you are, maybe you could talk a little bit more about the tools you're thinking to use for that, whether it's manual process or software or particular partners, or if you've got that figured out, or if you're considering options in that direction.
I think it's a little bit of both. I think we're using our registry where we're carefully cataloging genes and gene variants, and that will be one way that we match patients to trials. But I think even more importantly for now and for us, it's kind of looking at which sites in our network we think can most successfully complete whatever the trial might be, and which patients are close in location for those patients. I think our fear is that we're going to have to start prioritizing some clinical research over other research, because we just don't have enough patients to fill all of the trial interest. So we're internally going to have to figure out what we think is the most important path to go down for clinical trial development. And that's going to be tough. I don't have the answers, sorry, but I know what you're dealing with.
Dominique?
Hi, thanks so much for the presentation. I just wanted to add on to that comment or question that Paul had. In my prior role when I worked with the International Rett Syndrome Foundation, we built a clinical trial finder for our patient community that pulled not only clinical trials but also non-trial research opportunities. So whether it be survey-based or actually in clinic or virtual, no matter what it was. And so we would review them, list them on the clinical trial finder. And then that way our community had a one-stop shop, and it was agnostic to who was sponsoring these studies. It was really a way that we could let our community know about the research that was Rett syndrome specific. And we also launched a clinical trial committee. Because we kind of hit a point where we were worried we were hitting a saturation point. Too many trials that were simultaneously enrolling the same population within our community. And we did not want to get to a point that a trial did not meet its enrollment goals, and therefore a sponsor left. Because that would just send a really bad signal to everyone. And so through the clinical trial committee, we reviewed the protocols. We had patient representatives as part of that clinical trial committee, in addition to KOLs and our staff. And we would give guidance to the companies as to the likelihood of enrollment goals based on what else was going on at the current time. And so those were just some of the ways that we addressed those issues.
That's great input. So we've been in a position where until recently, there was no interest. Mm-hmm. So it's been, and I feel a little bit like we skipped the line. So there are no approved therapies for PCD at all. And now we're moving into gene-based therapies. So we didn't go through a lot of the other more symptom-based therapy trials. So this is very much a learning curve for us, but that sounds like a great idea to help not oversaturate the patient community.
Great. Is there any resource, Dominique, that you can share as far as, because I know for our rare disease, we also are oversaturating the market with clinical trials of the same population. So is there a contact at Rett syndrome or yourself that would be helpful maybe in that sort of survey development?
Yes. I'm no longer with the foundation because I'm now
Right.
at NCATS. But Melissa Kennedy, who is the CEO, and I can put her email in the chat. We very much felt like we learned from others. I was constantly tapping into other groups that I felt for any particular question were further along than we were as a foundation. And people were so generous in their time and resources that we always felt like we wanted to share what we had built with others so that we could learn from each other and all benefit.
Great.
Can I make one other quick comment that I forgot to make about benefits? And
Please. Yeah.
the Rett Foundation reminded me of this. It's not just that you get to know the people from the patient organizations. I have had the privilege of working with physicians like Alan Percy and people that would never have crossed my orbit except for because of this network. And that has just really been amazing. And you never know when those contacts will come to be very helpful in the future. So that's another benefit that I meant to mention and forgot.
Mhm.